response because he has connected, but unfortunately, he's not responding to our call. Yes, Deepyaman, we can see you. Yes. Dr. Deepyaman? Some issue at his end.
Yeah, some issue, it seems disconnected. Yeah.
So we'll wait for 2 more minutes, sir.
This is better than that. Okay.
Dr. Taruna,
Yes.
Dipeman has messaged that he is not able to hear anything. Some issue, it seems, for him.
So what do we do, sir?
I guess you also cannot hear me.
Yes, we cannot hear him, definitely. So can you ask him to join from another device? Ask him to try.
Yeah.
He's waiting in the lobby. It seems he's trying to join in from another device. Yeah. Dr. Deepyaman, can you hear us? Hello. Hello, Dr. Deepyaman. What is the issue?
Hello.
Yes.
Good morning, madam.
Dr. Deepyaman, is that Dr.
Yes,
Deepyaman?
yes, madam. This is Dr. D. Kumar.
Okay, great. Wonderful. So, shall we start the proceedings, sir?
You can start.
We are not able to see Dr. Deepyaman. Is it possible for him to put on his video?
More than 90.
We apologize for this connectivity issue with one of our participants from the organizing team. Dr. Deepyaman, can we hear from you? Should we start? Are you able to see my screen, Dr. Deepyaman? I think
I can see a slide here from the IICB. Is it there? You all are seeing that also?
Yeah, it is there. Slide is displayed.
Oh, yeah.
IICB slide is there, but
Although
his voice we are not able to
in
hear
the
him.
mobile, I can't read anything. It's there.
Now
And
you
I
are able
cannot
to see
on the
my
mobile.
see the flyer of the day.
Yeah, it has come now.
It has come now. Yeah. Dr.
And
Deepima,
the left,
should we go ahead? You are not able to join us. Is that? What should we do? Dr. Bala, can you speak to him, please, once again? And ask him, because we are getting delayed.
Yeah, the voice is also not clear in the mobile also. It's a lot of extra noise and disconnected suddenly. I won't be able to understand what is happening. Maybe signal problem somehow. I don't know.
Okay.
Yeah, twice I spoke to him.
So I think, Dr. Bala, may I request you to do the honors, or should I go ahead with introducing our chairperson?
Yeah, we will proceed. I'll take care of that. Yeah.
Okay then. So a very warm welcome to you all in this Indian Immunology Society Golden Jubilee online lecture series. And we have named it as Pratiraksha Swarna Mahotsav Vyakyan. So this is 7th of the series, and we welcome our esteemed speaker of the day, Professor V.R. Muthukaruppan, founder, director, research and advisor research, Arvind Medical Research Foundation, very actively working still. We fondly remember him as our founder member and former president, Indian Immunology Society, and former professor of immunology, Madurai Kamraj University. And we with a great welcome and warm welcome to Professor Nibriti Das, who accepted to be the chairperson for this session despite of her busy schedules and travel, who has been an anchor of the Indian Immunology Society, being a former secretary, former council member at IIS, and she has been contributing her services to the Nayati multi-super specialty hospitals in Mathura and in various leading roles and capacities. And she has been a former professor at Department of Biochemistry. I apologize for the error in the spellings, at All India Institute of Medical Sciences, New Delhi. And Professor Nibriti has been instrumental in bringing Indian Immunology Society to where we see it today. A lot of hard work from her side has not only been gone to the society activities, but also grooming their members. And now I hand over. This was a brief introduction for both our stars of the day, and I hand it over to Dr. Bala to introduce our chairperson formally for our budding immunologists. So, Dr. Bala, can you see the slide?
Yes. Yeah.
Kindly do the honors, sir. Yeah. Thank you.
It is my pleasure and privilege to introduce Professor Dr. Nibriti Das, formerly Professor of Biochemistry, All India Institute of Medical Sciences, New Delhi, former Member Secretary and Core Faculty, All India Institute of Medical Sciences, former faculty at AGU, Bahrain, former Director of Lab Services, Head of the Department, Lab Medicine and Biochemistry, and former Additional Dean, Nayati Multi-Super Speciality Hospital, Madura. Welcome you, Madam, for this meeting. And Madam Nibriti Das got PhD from the Department of Biochemistry, Lucknow University, and under the guidance of Professor G.G. Sanwal and under the mentorship of Professor P.S. Krishnan from 1970 to 76, and carried out postdoctoral research Associateship in ITRC Lucknow. And the area of expertise of Dr. Nibriti Das includes biochemistry, immunology, laboratory medicine, genetic and metabolic and immunological cardio biomarker analysis, molecular epidemiology, biochemical analysis and cell biology, biochemical and immunotoxicology of pesticides, laboratory medicine, complement proteins, and cytokines in systemic lupus, rheumatoid arthritis, and coronary artery disease, etc. as a potential biomarkers in therapeutic targets. Professor Nibriti Das, being a passionate teacher, mentor, and a counselor, pioneered the implementation of problem-based learning at All India Institute of Medical Science and imparted training to the residents, in-house faculty, and to other medical colleges through national and international workshops as an expert in PPL. She extensively involved in inspection and accreditation of UG and PG courses of medical institutions as a Medical Council of India appointed expert. Chairperson for framing the first competency-based syllabus for the MCI-driven UG biochemistry education, and more recently as chairperson appointed by NMC and reviewed and framed the new competency-based curriculum for PG biochemistry education. Can I have the next slide?
Yes. Just give me a moment, sir.
Yeah.
Yes, sir. No, the slide hasn't changed. Yeah, just give me a moment, sir. Today, is that okay now?
Yeah.
Okay,
And
great.
for the very important salient research activities and domains of Madam includes comprehensive studies on immune effector mechanisms in autoimmune disorders, revealed that modulation of complement protein C3D, complement regulators and receptors are closely linked with the pathophysiology of SLE, GN, and RA. And studies on free radical homeostasis, cell adhesion molecules, lipid profiles, and the genetic interlinks brought significant insights into the pathophysiology of coronary artery disease and hypertension in the Indian population, which is really a first of its kind research approach in Indian population and Indian medical context. Some gene variants of apolipoprotein like AIV, CE, ENOS, INOS, and peroxygenase considered protective in Western world are risk genes for Indians too. CR1 mutant allele associated significantly with the severe complications of Plasmodium falciparum malaria as higher level of CR1 expression that facilitate erythrocyte rosetting and microvascular blockade in malaria. Organochlorines, organophosphates, and pyrethroids derange macrophage functions, and active complement are also the areas of research for Dr. Nibriti Das. And she undertook UKRI projects, which is with Indo-UK collaborative projects with Professor Robert Moore's Department of Hematology, UOL, that is University of London, UK. The project developed a consortium with 10 clinicians and scientists from AIMS and 10 scientists from UOL to identify areas of expertise and common research priorities of both the institutions in the field of musculoskeletal disorders and infectious diseases. Next slide, please. And Madam has received a lot of awards, and prestigious and Mrs. Dr. G.P. Talwar Oration Award is a very prominent one among the awards she received from the Association of Clinical Biochemistry India in the year 2012. And first woman Indian to be elected as a council member for International Union of Immunological Societies, that is IIS, from the year 2016 to 19. Her initiatives as secretary, Indian Immunology Society, fetched the prestigious Best International Immunology Day or DOI campaign award from Professor Mantovani, President IIS, Professor Eva Kaufman, and Professor Joe Khalil from former presidents of IUIS. Only during the period of Nipriti Das Madam's as secretary, many youngsters really participated actively all over the country. She also, served as a chairperson or reviewer of the abstracts for the session Autoimmunity and invited speaker at the ICI IUIS International Union of Immunological Societies held at Melbourne, Australia in the year 2016, and a chairperson for the first IIS Forces Member Symposium held in USA. And also she was a member of General Assembly for IUIS from 2012 to 16. Next slide, please.
Yes.
So she's a member, core organizing team, Forces India, advanced course in basic and translational immunology held in Jaipur, March 2019. Best Teacher Award for Biochemistry by the Students' Union of All India Institute of Medical Sciences. Received, she was a member the New York Academy of Sciences, former member and chairperson, teaching committee of the Phase 1 Curriculum Committee and Staff Council of AIDS. Life member in several scientific national and international societies, including International Complement Society, which is very really highly prestigious society. She has published more than 110 publications to her credit in highly reputed international peer-reviewed journals. Next slide. Yeah, thank you. So once again, I welcome for the meeting. I request you, Madam, to take the session further forward. Thank you.
Thank you, Dr. Balaji. I'm audible. Can you hear me?
Yes, madam, we can hear you. Yes, madam, we can hear you.
1 minute. Yes. Thank you, Dr. Balakrishnan, for and practically I started listening to your introduction. And now I have got a very pleasant but difficult duty to introduce Professor Muthukurappan, a strong effort where I feel myself nothing but a small fry. And Dr. Muthukurappan today is
Yes, I am here.
in this book very often. It hasn't need any introduction. However, for the occasion, it is important that I introduce him.
Madam, Nibriti Madam, one please small request. Your voice is very so feeble. Can you please increase the volume if possible?
Okay. Can you?
Okay, madam. Thank you.
Can you?
I think.
Can you? Am I audible?
Ma'am, the voice is
Aruna, can you hear?
Ma'am, voice is breaking.
Switch on the video.
I think
Introduce
video switch off. Maybe that will.
Yes, madam. Switch off your video,
Yeah.
madam.
Can you hear me now?
Yes, better. Much better, madam. Yeah, please.
I switched off.
Yeah. Hope you are able to see the slides, madam.
Is it better? Okay. Now, it is a really good fortune for me to introduce Sir Muthukurappan, the founder member of Indian Immunology Society and as president. I remember that IIS previously honored him as the architect of the Immunology Society, and he was the recipient of honor the IIS, and first recipient had Professor G.P. Tamar. Am I audible? Can you hear me? Can anybody hear me? Can you hear me?
Yes, madam. Yes, madam. But only thing is, the voice is breaking. So if you allow me, I will just display these slides. Yeah.
Please switch on the video.
Because I have switched off.
Switch it on. We can't see
And
you.
Dr. Kefri, there is some connectivity issue at Dr. Nibriti Das's place, and that is why she had put her video. Yeah, because there is less bandwidth there. Yeah, she's joining from her mobile.
I can hear all of you. Can you
Great,
hear me
ma'am.
now?
Yes, much better, madam.
If you can't,
Yeah, we can.
if you can't hear me, I have no way out. Rather than Taruna, request you to introduce Professor Muthukurupan.
नहीं अभी बिल्कुल ठीक से सुन पा रहे हैं मैडम. अभी बिल्कुल ठीक से सुन पा रहे हैं. Please continue. जी.
Okay, fine.
जी.
So it is a real good fortune. Now सुन पा रहे हो?
हाँ बिल्कुल. बहुत अच्छे
stand
से.
up.
Yes, yes.
So Professor Muthukurupan, again I start with, he is a founder member of the Indian Immunology Society and past president of the society. Earlier the society had been honored by honoring him as the architect of Indian Immunology Society. I think it was in the year 1970, 2017. I don't exactly remember that, 2017. Professor Muthukurupan doesn't need any introduction, but a formal introduction is needed looking at the occasion to honor him properly. So, can I have the slides? Can you show me the slides? Professor Muthukurupan started his education. He completed his graduation and post-graduation at the Annamalai University. Taruna, the slides are not moving.
Ma'am, is it moved now? Yeah.
No.
No. Okay.
Next slide hasn't come.
Okay. I will just stop sharing and do the sharing again.
After his graduation, let me speak whatever I can without looking at the slide, and then you can put the slide. After his graduation from the Annamalai University, Professor Muthukurupan went to the University of Wisconsin to do his PhD, which he completed in 4 years' time. And then he continued there as a research assistant for another year. Then he came back here and joined the Madurai, I think, which Annamalai University as a faculty there. And time to time, he had several assignments in different colleges like Madurai University, Madurai Kamraj University, and then he went to the University of Wisconsin, USA, on invitation there as the visiting professor. Then he had been the professor of immunology, coming back again Madurai Kamraj University, and he had been coordinator on School of Biological Sciences. And presently, for last several years, he is the vice chancellor at the Bharati Dasan University, Trichy. And now at the moment, he is in the Aravind Medical Research Foundation, Madurai, and where he is continuing as the director of research. There are several. Next, Taruna.
Ji, ma'am.
Because I don't want to make any mistake just from my memory.
Ma'am, I've displayed. Hope you can see. Dr. Bala, has my slide moved? Yeah.
He has enormous contribution to the field of immunology with large number of research publications and PhD students that he has guided. So his previous interest had been in the phylogeny of immunity, immunology of leprosy, vaccines and immunodiagnostics, typhoid, and angiogenesis. Presently, his interest is pathogenic mechanism of diabetic retinopathy, corneal epithelial stem cells, pathogenic mechanism of uveitis, and biology and application of adult ocular stem cells. He has there is a list of huge number of honors and adulations that he has got. He has authored 3 books, and as I said, there are many publications. Next slide, please. Taruna.
Yes, madam. Yes, madam. Madam, can you
Awards
see?
and honors. I have now opened my laptop. Earlier it was not moving also. Can you hear me?
Yes, madam. Very well. Yeah.
Okay. He had been National Fellow of University Grants Commission, Fellow of Indian National Science Academy, recipient of Ranbaxy Research Award for Basic Medical Sciences, Fellow of Indian Academy of Sciences, recipient of Swami Pranaveshananda Award for Senior Scientist on Developmental Biology, and recipient of National Institute of Immunology as the Senior Scientist Award of the Indian Immunology Society. These are just few of many awards that he has received, and the way he is acclaimed in the international and national seniority of the immunologists. So, any more? Perhaps not, Taruna?
Grateful, madam. You can now request Professor BRM. Yeah.
So this is a very brief account of Professor Muthukurappan's introduction, and I very sincerely welcome Professor Muthukurappan and extend my good morning wishes. And it is a privilege for me, rather I will say it is a very good fortune for me, a small pie like me, to invite Professor Muthukurappan to share his work and wisdom gathering here. So Professor Muthukurappan, please grace us with your presentation. Is that okay?
Sure.
Taruna?
Yes, madam. Yes, madam. We can see his slides now. Yes, sir. We can see your slides.
All the members of the Indian Immunology Society and all the others for attending this lecture. Thank you for coming. And Indian Immunology Society President, Professor Madan, thank you so much for organizing such a very interesting Jubilee lecture. And also, I thank you for inviting to present the 7th lecture. Lion, thank you so much. And, as mentioned already by Dr. Das, I was a founder member of Indian Immunology Society, along with Professor G.P. Talwar, Shobha Sehgal, and Dr. Malavia, and others. And the members of the Indian Immunology Society gave me the opportunity to serve as the president of the society, 81 to 82. In all these days, the annual conference, I enjoyed visiting and discussing with the young immunologists. That was always, I remember my interaction with the members of the Indian Immunology Society. And our secretary, Dr. Darshan, has suggested that I should talk about my first series of research project that's about the phylogenetic community, and that refers to the ontogeny of B lymphocytes in the lizard. And as Dr. Balakrishnan is popularly known as in Madurai Kamaraj University as Immuno Bala, and we will call him also Immuno Bala. Thank you for organizing this.
Thank you, sir.
Now, when I want to talk about the phylogeny of immunity and my research work, there is a next slide. Now we will have to go back to the beginning of the modern immunology and what's happening during that period. And now, as you can see on the left, as early as 1953, the acquired immunological tolerance was demonstrated by immunomadaver in the mouse by transferring the lymphocytes from another strain, inducing tolerance in that. Subsequently, in the University of Wisconsin, Dr. Ray Owen about the dizygotic twins, and they share the common placenta. Accordingly, red cell chimerism is developed accordingly, and the twin members will be able to tolerate the allografts from another sheep of the same twins. And then the most important event to take place is by McFall and Burnett in Australia is the clonal selection theory that forms the theoretical basis for the entire immunology to be followed. And then the most important thing is the discovery of the thymic function in London by Miller, and 1962 in University of Wisconsin, the function of the thymus was discovered by Wolf. And then at the same time, when I was a student there from 1961 to 65 in University of Wisconsin, Dr. Robert Auerbach, my professor and mentor, in 1962, he has demonstrated that the lymphocytes appear first in the embryo in the thymus, and that was the most significant event also. And Robert Good also, the humans showed there are diseases to demonstrate a dichotomy of the immune system, the T and B cells. There are diseases affecting the T cell function, diseases affecting the B cell function. And 1968, NASL in Australia has demonstrated one B cell produces antibody of only one specificity, and several people were involved to demonstrate the T helper cell and T cell B cell lymphocyte interactions. Cooperation is required. And Edelman and Porter have described the chemical structure of the antibody, and Jean Dose in 1980 explained the presence of the major histocompatibility complex in humans. And the Milestone and Kohler have shown the way of developing the monoclonal antibody in 1964. And in the same year, people have described the antigen processing and presentation. And 87, the generation of antibody diversity by Susumu Tonegawa. And then the major event is 96 is the Dougherty-Single phenomenon. And as you can see, in the whole beginning of the modern immunology, several people have received the Nobel Prize. For example, Burnett, Edelman Porter, and Jean Dose, Kohler and Milestone, Susumu Tonegawa, and Dougherty-Single. And this I learned, during my visit the whole field of immunology was introduced to me. I learned immunology, even though I went there to study the developmental biology. I also did my project on development of the lens in culture, and that finding that tissue interaction between the epithelium.
Sir, excuse me, sir. The slides are not moving, sir. It is a birth of yeah. Now it is moving. Okay.
Okay, thank you.
Right, sir.
Thank you. I am when then by the time I was leaving, I applied for a project to National Institute of Health. The project was sanctioned. By the time I returned and established back in Annamalai University, and the project was research project of my study was the to study the immune system of the lizard to elucidate the origin of B lymphocytes, and that's mainly the question of asked was the major question asked was the dichotomy of the immune system, whether it is there during evolution of the immune system, and what is the bursa equivalent. Bursa is only in birds, where what is the equivalent of the bursa in mammals, and that was the main question asked in that project. And I had 28 students did PhD with me on the lizard immunology, and 9 students received PhD on the lizard immunology, 28 papers on this in the lizard in various journals, and 69 to 80. And these are the titles of my PhD students on immunology, lizard immunology. And at the time when we started, when nobody knows about many things about the lizard, the garden lizard, how to keep them in the university animal house, how to maintain them, and this the housing for the lizard. You can see the lizards house, and also how to feed them, but they will eat only the moving insects. So this way we maintain the lizard for several months in the laboratory when the immunology experiments are being carried out. And in addition, we will have to also understand the development of the lizard. And we could collect the eggs of the gill, and they incubate the eggs in the wet sand in the laboratory at 30 degrees centigrade. And every 2-3 days, we will collect the eggs and identify the various stages of the lizard. As you can see, this it looks very similar towards chicken and then various characteristics. It is possible each of these stages refer back and learn about the development of the immune system during the various stages of the lizard. And in the lizard, there are primary lymphoid organs, thymus and the liver, and secondary lymphoid organs, bone marrow, spleen, and there is no well-organized gulf. It is only a loose aggregate of lymphocytes. There is no bursa, no lymph nodes, and no tonsil, no pairs, patches, no appendix. And we will have to develop our own methods to do various surgery and thymectomy, splenectomy, how to engrafting, and whether we can do serial bleeding, and it is possible to do serial bleeding using the retroorbital sinus as in the mouse, and to understand the immunoglobulins, antibodies, purification of immunoglobulins, and the action of antibodies to IgM and IgY of the lizard, and also to identify by immunofluorescence microscopy T and B This is the allograft rejection, and you can see the autograft after 2-3 weeks, and the allograft, the changes taking place. One can see the normal blood vessels and also the melanophores, and those melanophores are all disintegrating slowly after 24 days. After 45 days, the whole skin is rejected, as you can see in here, by 45 days. And allograft rejection has been demonstrated. And then we learned about the immune response to sheep erythrocytes. That's one of the common cellular antigens used in mice, and we use the same. And also we have established the Cunningham-Sandberg experiment of the plaque forming cell assay, the PFC as popularly known as, and this is the lymphocyte secreting the antibody, and in the presence of the complement, all these red cells are all completely cytolized, so that you can see a plaque. And this is a very sensitive technique, looking at the plaque forming cells in the various tissues. It's possible, and this reaction is very specific. Instead of after immunization with the SRBC, and if you put in this test system, a rabbit RBC, there won't be any lysis. That antibodies are specific to the SRBC. And one can also follow the various stages of PFC response to SRBC. After immunization, there is a small lag phase, and there is an immune response peaking about 2 weeks. And when the second immunization is given, and this with the SRBC, there is very little of lag phase, but the PFC response is not very higher to the primary response. Then we also looked at the antibodies, serum antibody response, and subsequent to PFC formation. We can see the formation of the antibody response in the primary response after second immunization, secondary response seen in the result. Then we looked at how early the antibody response to SRBC can be found, and we incubated the eggs, and on the weight we could follow the 12 hours after hatching. This very young, this and at 12 hours after hatching, there are PFCs in the spleen, and 1 day also is present there, and we can follow in various stages, and in case of the control, there is no PFC in the spleen. And then to look at the role of the thymus in the allograft rejection and what is their function. So we learned that the thymus is present in very close to the internal jugular vein, and there are 2 lobes, 1 pair in each side. These thymic lobes, you will have to do the bilateral thymectomy to remove the thymus tissues, thymus gland. And then we carried out experiment of thymectomy, we immunized, and we to look at long-term effect of the adult thymectomy in SRBC. In this sham thymectomy, see, we showed a formalin fixed SRBC that will the for function and the T cell. And in the thymectomy, you can see the PFC response as cells as much as by 6. In the case of the thymectomy, very drastically, very significantly reduced after thymectomy when immunized, they get reduced. So demonstrating the of the thymus T cell response in the anti-SRBC response. Then we go to the bilateral thymectomy, thymic dependence of PFC response to SRBC. To summarize findings, we carried out a bilateral thymectomy. We have also after thymectomy, anti into and we found that the anti-thymocyte serum abrogates the anti-S response, and also 1 month after thymectomy, the PF's response is abrogated, and it is important to see the recovery of this PF's response occurs only in the presence of the thymus. So if you, instead of a thymectomy, if you carry out sham thymectomy and then anti-thymocyte serum, they recover the T cell response after some time. Now we have identified the IgM in the lizard, and that's similar to human IgM in terms of the molecular weight of the heavy and light chain, and also the lizard possesses the 2MA resistant in contrast to the IgM, which is 2MA sensitive, 2 mercaptophenol sensitive, low molecular weight, and this is similar to a bird immunoglobulin, and we so called it IgY in the lizard. We have raised very specific antibody in the rabbit for lizard IgM and lizard IgY. And then we can look at the surface IgM positive cells during various tissues in the lizard, and immunofluorescence microscopy was used. And first we treat with the anti-IgM and followed by the secondary antibody, anti-rabbit FITC, so that one can see the surface IgM positive cells as the fluorescent cells. Such cells are present in the spleen, this much, and blood, bone marrow, thymus, almost nothing. So we have also got the Ig mu, anti-Ig mu. We have also used Ig epsilon, IgY antibody to look at this, and they are low, and you can see the high level in the spleen and blood. And then in order to learn about the origin of the B cells in the lizard embryo, we looked at the various stages in the liver, the histological picture of the liver showing the liver parenchyma and the lymphocytes, and this is at stage 34. Stage 34, and you can see these lymphocytes in the liver at various stages. In 33, such cells were absent. In 34, you can see such cells as much as the number of lymphocytes per liver, about 7,000, and it goes up and peaking at the stage of 36, and then it goes down in the adult. So, surface IgM positive B cells in the lizard. In other words, in order to see whether these surface IgM positive cells are the B cells, we use a system of cycloosphosphamide, and B cells are the mammalian B cells are sensitive to cyclophosphamide, and we looked at the surface IgM positive cells in the bone marrow, in the blood, in the spleen. After cyclophosphamide treatment, such cells are reduced in the bone marrow. Same thing is in the blood and also in the spleen. But 10 days after COI, such B cells generated in the spleen, regenerated in the spleen. And this clearly demonstrating that surface IgM positive cells are the B cells. So look at the antigeny of the B cells. So many cells in the spleen and peripheral blood and bone marrow, they are all but 1% only surface IgM positive cells. And after thymectomy, as expected, the such cells, surface IgM positive B cells are increased in the blood and spleen. And surface IgM positive B cells appeared first in the stage 4 of the lizard embryo. Therefore, the embryonic liver is the site of origin of development of B cells in the lizard. Maybe we can say liver in the lizard is a bursa equivalent, but in the adult lizard, it is not having, and it may be the bone marrow. Now, in order to learn a little more about the phylogeny of humanity, I did a little bit of work on the angiogenesis. I'm sorry, a little bit of work of the immunology of the fish. Sorry, slides are missing. The two students did PhD on the immunology of the fish. We have also learned many things in this fish. We use the teleost fish, Tilapia mosambica. The first thesis we have developed, my student did in 1974, is really somebody from Europe wanted to see the thesis. So I sent to this person, and somebody else again in Europe asked for the thesis. Then I requested the first person to second person, and subsequently second to third person. That thesis was circulating in Europe, very interesting at the time, because many of the things, whatever it is, we have done the fish immunology for the first time. Then little bit I will spend about very brief time on.
Sorry to interrupt you, sir. Your slides are not moving.
Yeah.
Thank you, sir. Yes, sir. Yes, sir.
Very briefly, I would like to do some very significant work I did on angiogenesis, and I was able to go to University of Wisconsin as visiting professor for 1975. For 1978 to 79, and then again in 19s. And during that period in Bob Arbach's lab, I was able to do the angiogenesis work. And first, let me define the angiogenesis. It's a process of new blood vessel formation from the existing blood capillaries. There will be permeability of the endothelial cells, and the endothelial cells proliferate and migrate, and remodeling and expansion of the vascular network occurring in the embryo and even in the adult. There are two types of angiogenesis. One is the physiological angiogenesis, as occurring during development and wound healing, regeneration, especially during the placenta development. And also, in the case of the pathological angiogenesis, it is occurring during inflammatory reaction, in the allograft rejection, in the tumor growth, in the also connected with my present work in the Aravind Hospital, diabetic retinopathy and Eales disease. In these two diseases, there is overproduction of the angiogenesis, new blood vessels in the retina, causing the disease. Judah Folkman has pioneering work has done about the tumor-induced angiogenesis, and she explained that for the tumors require blood vessel supply, only then it can grow. Solid tumors occurs only when it is vascularized, and he postulated that tumor secretes a factor, and that factor is responsible to attract the growth of the blood vessels towards the tumor so that the tumor can be vasculated. And um, in the absence of the vascularization of the tumor, tumor does not grow beyond 1 to 2 millimeter in diameter. And when Folkman said all these things, there is a big controversy among the medical profession that vascularization of the tumor is not due to any factor. It is only an inflammatory response to the Tet cells. There are no factor for the formation of the blood vessels from the tumor. So they were against this thing, and at that time, Folkman and others used in 74 rabbit corneal model, and this is possible to do the mouse tumor in the rabbit cornea. It's an allogenic combination. In the chicken also, it is very difficult to distinguish the old blood vessels from the newly formed blood vessels. And under this condition, so I have set up to develop this model in the mouse for tumor-induced angiogenesis, and the importance of mouse corneal model, the inbred mouse strains and syngeneic tumor can be used. This will not involve inflammatory response, and it is possible to observe the new blood vessels sequentially in the mouse cornea. The newly formed vessels can be quantified also in the avascular cornea. And this is a micro surgery. That's my hand, actually, you see, to implant a tumor into the corneal stroma. And we can make use of the C57BL6 and C55 mammary adenocarcinomas in a syngeneic combination. And if you use the biopsy and the CSO90 tumor, it is allogenic combination. And we have to have the fiber optic lighting. This lighting will not produce any heat. If you have such a light with producing heat, that itself will induce inflammation and new blood vessels. So we'll have to avoid that. And I will have to develop my own surgical iris spatula. Since one will have to make a pocket in the stroma in the cornea, which is 1/10 of a millimeter thickness, and it is possible to do this in the mouse and implant about 0.5 millimeter cube tumor into the stromal pocket and follow the mouse. And as you can see, this is the location 1 millimeter away from the limbus, which is having the blood vessels. This cornea is no vascularization at all, and that must be clear. And this is the tumor cell implanted 6 days later. You can see the new blood vessels going, very directed growth of blood vessels towards the tumor. We call this brush powder because that is a place where the endothelial cells are proliferating and leakage, so that there will be extravasation of the blood cells. And 25 days later, that tumor is vascularized, and 3D dimensional growth of the tumor possible only after vascularization. And in the syngeneic combination, in this case, the tumor is growing within the cornea. This is the corneal epithelium, corneal stroma, the tumor cells. And as you can see, the new blood vessels in the cornea, and there is no infiltration of lymphocytes or mononuclear cells, and it is very clear in the syngeneic combination, biopsy mouse 180 tumor. But on the other hand, in the allogenic combination, along with the tumor cells, you can see the large number of infiltrating. This is the allogenic combination, so that tumor can grow in the absence of infiltration, and tumor-induced angiogenesis has been very clearly demonstrated in the absence of infiltration or inflammatory response. And subsequently, summarizing this study, tumor-induced angiogenesis not due to inflammatory response, contradicting then the existing contention that tumor vasculature was an inflammatory response to dying necrotic tumor cells. So tumor-induced angiogenesis is due to a diffusible factor from tumor growth. I will have to prove this point in the next slide. Then subsequently to my study, people have demonstrated by molecular biology techniques, they have developed, they identified that the factor from the tumor is vascular endothelial graft factor called VEGF, and they have also generated the recombinant human VEGF was made available, that's commercially available for the study. So during my next visit, I repeated the same experiment. Instead of the tumor cell, I used the vascular endothelial growth factor, and I used the slow release polymer and incorporated the known amount of VEGF in the slow release polymer and implant 1 millimeter away from the limbus in the mouse. And as you can see, even 4 days later, you can see the new formation of these blood vessels, and these are all in the background. They are all the iris blood vessels. And after 9 days, the VEGF bearing graft is vascularized. After exhaustion of the VEGF, all these blood vessels will regress very clearly, and this very clearly demonstrated instead of tumor, VEGF by itself can induce the formation of the new blood vessels. And these two papers, tumor-induced angiogenesis and also VEGF-based angiogenesis, are published very well in the well-known journals. This is in American Journal of Ophthalmology 1993 published. Not only the paper was published, the editor wanted to publish this as a cover picture. So the American Journal of Ophthalmology, my own study was published as the cover picture showing the graft with the VEGF and the new blood vessels, and that comes to an end that tumor-induced angiogenesis is due to VEGF. And whatever I did during this period, if now also be meaningful to learn more about the angiogenesis in the retina of the patients. And in the case of diabetic retinopathy and Eales disease, we are able to do much work on understanding whether the VEGF is involved in this process or not. Thank you very much. Now, sorry, the next slide is not moving. Thank you. Now I joined Aravind Eye Hospital, Aravind Eye Care System about 23 years ago. And at the time, the founder chairman, Dr. G. Venkataswamy, all the time used to say, intelligence and capabilities are not enough. It's not enough just to have a fellowship or salary, but there must be a joy of doing something beautiful. And this is what we follow for all my students and also the present work in Aravind Eye Hospital. I would like to refer to him as a great man. 23 years I joined as a director, organized research in Aravind Eye Care System, and I am continuing now as the advisor to research. Thank you very much. I am very much thankful to the president of the Indian Immunology Society, Dr. Professor Mother, for giving me such opportunity. I will be happy to interact for any questions.
Thank you, Professor Madhukar. We are really grateful to have heard this great talk from you. And it has been very educative and learning experience even at this age. And I think everybody is benefited by your research and the approaches of the research that you have taken. And your end message is the most valuable also particularly for the budding researchers. So with this, first, I will also thank all the audience here who have graced this occasion with their patients and
प्रतीक है
also
बुला इधर पानी का अच्छा ठीक है नहीं मत बुला मत
and also initiate the interactions with professor to all the participants. So,
Dr. Madan, I want
questions in the chat box. Thank you so much.
just 1 second, Dr. Madan. I want
yes sir
Auerbach, my major professor.
yes sir yes sir we honor Professor Auerbach and thank you so much at so late in the night past I think 2 hours past midnight and we are really really grateful for your presence sir you have motivated all our youngsters to do some great things in life and we definitely thank you for giving us Professor VRM in the form that he's today. He still continues to carry forward your work and both of you did a great service to humanity by showing that evidence that the tumor is in fact inducing angiogenesis on its own and it is not an inflammation induced reaction. Thank you so much, sir. If you would like to speak something, we would be honored, sir.
Can you hear me?
Yes, very well, sir.
Well, I want to congratulate Professor Muthukurupan on a marvelous career. He has been an absolute delight and has done about the most exciting things you could ask for in a laboratory. And every time he came to visit, he did something that got published in major journals and became used, like the corneal eye graft, for example. He forgot to mention that his PhD thesis actually involved the study of the lens of the eye. And going to Aravind is, in a sense, a return to his first love. Anyway, congratulations.
Yeah, really incredible model to show the thing. May I ask you both how this idea came to you to use the cornea implant for showing the tumor angiogenesis? That would be very interesting for all of us to know.
Yes, that's really true. And now this, when I was there in University of Wisconsin as visiting professor again, and we wanted to study, Bob Auerbach has been there in Folkman's lab and learned about angiogenesis. And so we wanted to study in a system where there is no blood vessels. There are whether there we can do in syngenic combination of the tumor and the mouse, and that's possible only in the mouse cornea. So at the time, the people were doing that only the corneal model in the rabbit, but in the mouse, it is only 0.1/10 of a millimeter thickness. Is it possible to do surgery with such a thin cornea in the mouse? That was the first challenge. And somehow my hands are very steady, capable of doing this surgery and implant a small piece of tumor within the stroma. We'll have to do it within the stroma. If you injure the epithelium, that itself induces angiogenesis. One will have to try. With the confidence, I was able to do that. And in the syngenic combination and allogenic combination. At the time, Bob Auerbach also was studying angiogenesis induced by the lymphocytes, allogenic lymphocytes. And lymphocyte-induced angiogenesis, he was interested at the time. So I was able to do the lymphocyte-induced angiogenesis also. The mechanisms of angiogenesis are different in these two situations. And as I showed, the tumor-induced angiogenesis, profuse growth, and brush powder but in the lymphocyte-induced angiogenesis, the blood vessels grow, but there is no leakage. There will be proliferation of endothelial cells. There is no leakage. And how they distinguish lymphocyte-induced angiogenesis and tumor-induced angiogenesis. This was first published in the well-known journal in USA, Science. And then subsequently in another cover picture of whatever I have done. Okay, thank you.
I think in terms of your question, the original method was in the rabbit.
Yes.
Was very useful, but you could do maybe 6 rabbits. You had no genetic control. You had no tumors to work with except 1 or 2. And the moving it to the mouse allowed all the genetics to be done and to do reasonable immunology with it. And so that method became sort of what they called the gold method
Yes.
for studying angiogenesis. Much is done in vitro, but then you always want to go back into an animal system. And this is the way that worked. And ultimately, that was useful in the development, as Dr. Muthukurupan mentioned, once the VEGF situation was developed, then the mouse became the assay system that demonstrated things like Avastin, treatments that are now used in cancer treatments.
Thank you, Professor Alva. Professor Nibriti, may I ask a question to Professor VRM?
Yes.
Yes.
Sure.
Yeah. Thank you so much, ma'am.
I have had it over capacity. Okay.
Sure, ma'am. Sir, why adult thymectomy? When you were showing the lizard immunology, summary, you had mentioned adult thymectomy led to increased number of B cells.
Yeah.
A very intriguing statement for me. Can you please elaborate on this?
The spleen and the blood, lymphocytes origins, thymic origin, and the liver origin. T and B cells should be present. Once you remove the thymus, no more T cells are coming into circulation in the spleen. Consequently, the percentage of T cells in the total lymphocytes is increased. That's why the number, all right, that's what's happening. So in the absence of the T cells, whatever you see, B cells, that's what's happening. Thank you.
Thank you, sir. I can see certain comments for you, sir, from various participants and our audiences. So Dr. Soma Mandal says, thank you, sir. I grew up reading your papers. Dr. Manoj Kumar says, thank you. I think Dr. Deepyaman is joined us from there. And Dr. Balakrishnan says, very interesting and elaborate experimental approaches to learn immunology. Thank you, sir. Dr. S. Ayub is saying, thank you, sir, for your lecture. Dr. Rashmi Singh says, thank you for your wonderful talk. Dr. Krishna Prasad, formerly at AIMS Department of Biotechnology, says, Professor Muthukurupan, greetings. Always was impressed as a student of your group's work on B cell ontogeny using lizards. Dr. Rajni Rani is commenting, very interesting talk. Thank you so much, sir. And Professor Krishna Prasad again writes, we miss your presentation related to leprosy and typhoid. Warm regards and best wishes. And Vignesh Sundarajan writes, very inspiring, sir. Thank you very much. Professor Shubhada Chuplinkar, our former director, ACTREC, writes, thank you, Dr. Muthukurupan. Pleasure to see you after a long time and hearing about your fantastic scientific journey. Very inspiring and best wishes.
Thank you. Thank you. Thank you, everybody.
Yes, sir. So we will propose a formal vote of thanks. And after the vote of thanks, we will be still connected for any informal chat by any of the members. That is, yes, sir.
For vote of thanks. Just
Yes, sir.
2 sentences about the leprosy wall.
Most welcome, sir.
It is well known that the polar leprosy, lepromatous, and the tuberculoid among the two, the lepromatous leprosy, the T cell response is very much reduced. Cells are not and they have very clearly demonstrated that mechanism by which the T cells are not responding and the immune response to T cell is reduced in the lepromatous leprosy. Polyclonal activation is what we have demonstrated using the cells of the lepromatous leprosy. There is a receptor in the lymphocyte T cells called E receptor, and that E receptor and that application Dharmendra preparation of the leprosy bacilli, we can incubate and show very clearly cytometer that the E receptor is modified and they are not able to respond to mitogens and antigens. Thank you. Thank you. Thank you.
Very grateful, sir. And I will now share our slide for a formal vote of thanks. And I invite Dr. Aparanji. Hope my slide is visible, Dr. Bala.
Yes, yes.
Thank you, sir. Thank you. Dr. Aparanji.
Yes. Good afternoon, everyone. Good afternoon, everyone. Can you hear me?
Yes, 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 Dr. Aparanji.
Okay. On behalf of Executive Committee of Indian Immunology Society, with immense gratitude, we express sincere and wholehearted thanks to our eminent speaker, Professor V.R. Muthukurappan, former President IIS and Advisor Research, Aravind Medical Research Foundation, Madurai, for the prestigious 7th IIS Golden Jubilee Lecture and sharing highly valuable data on the basic immunological techniques, including the ontogeny of B lymphocytes in reptiles, especially in at various stages of developmental stages, the lizards, immune responses. And sir, it was really an interesting and inspiring journey for us. We are indeed honored by your presence and sharing of your rich experience in the field of basic immunology and angiogenesis and corneal epithelial adult ocular stem cells. And we express our heartiest thanks to our esteemed chairperson of this event, Professor Nibriti Das, former professor of biochemistry, AIIMS, New Delhi, former secretary, IIS, who worked tirelessly for the growth of the society, for having accepted and orchestrated this event in the most scientific way. We thank all the participants across the country for their overwhelming response. Thank you all. We place and record our sincere thanks to the IT team, ICMR, NIRRCH for the seamless work in making this event a grand success. Thank you one and all.
Grateful, Dr. Aparanji. And over to you, Dr. Uma Kanga. Dr. Uma Kanga, can you hear me?
Hello.
Yes, Umaji.
Good
Yes.
morning, everyone. First of all, sir, thank you so much for the wonderful lecture. We really appreciate and we've learned a lot, and our gratitude to you for sparing time and educating us on this. Sir, my humble submission that we are having this immunology conference, and this is a Golden Jubilee event of our society. You have been a former president of this society. So I'm taking this opportunity to convey to you that we would like to invite you at the conference, and we want all former presidents to be present during our inaugural function. That is our wish and our dream. And we request you to kindly accept this invitation. We will send you formal letters, sir. But I thought this is a good opportunity for me to personally convey to you on behalf of the whole organizing committee of the conference that it will be an honor, sir, that you will join us at the inaugural function. All former presidents would be present, and we would need your blessings that we are able to do the conference successfully. So we will send you formal invitations.
And this invitation stands true for all our participants who have joined us today, that please join us in Immunocon 50 and with the organizing committee, which is headed by Dr. Uma Kanga, who is the convener and organizing secretary, and Dr. Sujata Mohanty, who is our co-convener. We are launching our website today, sir, and wishing you all a very successful Immunocon 50 from IIS. Thank you. Thank you, Dr. Uma.
I would like everyone to please encourage all the students in the lab to submit your abstracts. The call for abstracts will open today or tomorrow. The website will be live. And please encourage your students and your colleagues to support the conference, attend. And I'm sure we are putting in all the efforts, and I'm sure you will all enjoy the scientific content in this conference. Thank you, Taruna, for giving me an opportunity to give an invitation over here to all of them together. And I will look forward to seeing a lot of abstracts, and we are waiting for you all to be in Delhi.
Before
yes, madam.
we end, I will like to have a word.
Sure, ma'am, please. We are not going to end now. We are going to have an informal chat with all the members. Yes, ma'am.
तो मेरे को देखो अभी मैं
Uma ji, okay, yeah, please go ahead, madam. Yeah. Yes, Nivriti ma'am. Yes, madam, please.
I wanted to extend my sincere thanks to you, the president of IIS, and the team IIS to have organized this wonderful talk by Professor Muthukarupan that we will always remember very, very gratefully. And also the vote of thanks, again thanking me to Aparanji and Dr. Bala to have introduced me, and the team IIS to be here to enjoy, to make me enjoy this event, also to have selected me as a chairperson to be here with all of you and Dr. Bala. So all the best wishes to Uma and all of you for the forthcoming conference. Thank you again.
Thank you for your blessings, ma'am. And Professor Arva, would you like to say something more to the youngsters who have joined us today? Yes, sir.
I just want to say that science is exciting, and one has to have fun as well when one is working in the lab.
Yes.
Nothing more exciting than finding new things.
It's not audible.
Something that's
I
worth
couldn't hear anything.
Am I muted?
No, sir. You are able to. We are able to hear you. Please continue. Yeah. Please continue, sir. Please continue, Professor Arva.
I think he has muted his call. I think.
Yeah, thank you. I will just see
Now
that.
you can hear me?
Yes, very well, sir. Yes, sir.
I think I've said what I want to say. It's just science is wonderful, exciting, and it's nice to have young people join because the work continues. We learn more all the time.
Absolutely, sir. Thank you so much.
I congratulate the whole Indian Society of Immunology. I've seen it grow from nothing to being a big organization, and people who are here demonstrate it. Congratulations to everybody.
Thank you. Thank you, sir. We are really having a proud lineage and we are following the footsteps that our elders have shown us. And very happy to inform you, we have 1900 plus members of the society. And we had Immunology Day celebrations across the nation, more than 50 at more than 50 centers. So it was a great celebration this time. Thank you. May I request KP sir, if you would like to speak to VRM sir, Professor Krishna Prasad, Dr. Chiplunkar.
Yeah.
Yes, madam.
No, it was a pleasure listening to Dr. Muthukurupan and to Professor Orbach and to hear their journey. And I want to tell I was just looking through the bio sketch of Dr. Orbach now, and he plays piano, and he's taken a lot of interest now in playing the piano. So I wanted to ask him, how does he spend his time between doing science and playing the piano?
Easy. Like when I was a student, I could work in the day and then play piano at night.
Oh, great. That's nice.
The piano.
Oh, yes. There, it's next to him. Yeah. Thank you. Thank you, Dr. Muthukurupan. It's nice to see you after such a long time.
Thank you.
And I hope we meet at the conference in Delhi.
Thank you.
Yeah. Thank you. Thank you so much, ma'am. KP sir, can you hear us?
Yes, yes.
Please.
Hello, sir.
Hello.
Nice to see you, sir, and nice to hear your presentation.
Thank you.
I remember the earlier immunology conferences we used to attend as students, and I used to admire the fact that you could use these reptiles and lizards for studying immunology. And I also recall your slides on the spleen assays, antibody forming cells. It was wonderful. And that time your great emphasis was on humoral immunity.
Okay.
It was really wonderful. And I thank you for giving us this opportunity. And I wish you all the best.
Yeah. Thank you.
May I invite anyone else who would like to speak to the chairperson, to our guest of the day and our speaker of the day, Professor VRM? Please feel free. Okay. Are we hearing from anyone? No, I don't think so. So with this, we come to a close, sir, and a sincere gratitude from all of us at Indian Immunology Society to Professor VRM, to Professor Nibriti Das, and Professor Arbak for joining us today, and to all our esteemed participants who spared their valuable time to join us today. Thank you very much. And this talk would be available on YouTube all the time for anyone to hear it again. Thank you so much.
Thank you. Bye-bye.
Thank you.
Thank you. Bye, Taruna. Thank you so much.
Bye-bye.
Bye-bye. Thank you. Thank you.
Nice to see you, Bala. Nice